and then just a little bit of my undergraduate background so you guys can ask your questions and stuff. So I won't take up too much time with this part. So the Mind Network is something that I started with a couple other graduate students about a year and a half ago, maybe two now, with the purpose of establishing a network of faculty, graduate students, and undergraduates who all like studying the brain, and just to kind of create a nice safe space for everybody, and also to help foster some of those mentoring relationships. So you can seek advice, so you can have like somebody. So the biggest part of our network is the mentoring aspect of it, where we pair a student, so either a graduate student or undergraduate student, with a faculty member or a graduate student in the case of an undergrad. And that gives you like a person as that kind of acts as your personal support system. So if you need somebody who's already studying a topic that you're interested in, to kind of ask questions about how they did what they do, how they got to where they are, that's the purpose of the mentoring aspect of it. In addition to that, we also host events where we bring in speakers and we ask them to not just talk about, the science that we all love, but we also ask them to talk a little bit about their background and advice for, how to navigate the field as a minority student. So that's the network in a nutshell. So if you guys have any other questions about it, I'll be glad to answer them after this part. So in addition to that, I'm a graduate student in the lab of Dr. McGann, as Hannah said. And what we study in our lab is sensory perception. So I'm gonna actually share my screen with you guys really quickly. So can you guys see the presentation?
Yep.
So just to kind of orient you guys on the olfactory, so use the olfactory system as a model. And what happens typically when we smell something is we have these olfactory sensory neuron cilia that sit. Can you see my pointer also? Yeah. Okay. So we have these olfactory cilia that have all these receptors that are specific to different odorants. So everything that we can smell has a specific receptor that lies on these cilia. So you can think of these as kind of the dendrites of the olfactory sensory neurons. And then we have the cell bodies that sit just inside of the olfactory epithelium. So once something binds then to the receptor, then that signal is then sent back through the axon to the glomerular layer of the olfactory bulb, where we have some early processing taking place. So as you can see, this is where the OSN's axons terminate, as well as connect with the primary output mitral tufted cells. We also have these periglomerular cells that are GABAergic interneurons, and we have these GABAergic and dopaminergic short axon cells that also process but they alter the signal to kind of quiet down glomeruli that are nearby in order to kind of sharpen the signal that we're getting. So now that we're oriented, and then in addition to that, we have all these neurotransmitters coming into the olfactory bulb. So I like to start, before I start explaining my project, usually by presenting this picture that's very, very old. This is one of Golgi's first stains of a dog olfactory bulb. And just like, it looks just like the anatomy I just showed you, where we have the mitral cell layer here. And then if we go deeper, we have the granule cell layer. These are our glomeruli. These are probably periglomerular cells or short axon cells. It's kind of hard to tell from this picture. But the thing I want to draw your attention to is, so we have all these inputs into the olfactory bulb from elsewhere, from outside, so from those OSNs. But in addition to that, we have all these lines here that come from basically everywhere else in the brain that terminate at the level of the olfactory bulb. So what this opens us up to is a rethinking of our peripheral sensory systems, right? So we think about, memory and learning as happening, has to happen in the hippocampus, has to happen in the neocortex. However, since there's so much input from other brain regions to the olfactory bulb, it's very possible that there are changes occurring even at this very, very early stage. And if you think about it, um, if I have a warning signal, if I establish a warning flag very early in my system, then my reaction time can be much shorter than if I had to actually send that signal back to the higher brain regions for it to be processed, and then I'm able to react after, and then that signal's then relayed back down, and I'm able to respond. So it actually makes a lot of sense when you think about, defense mechanisms and moving and avoiding danger. So previous work in our lab has actually done some discriminative fear conditioning. I'm not sure how familiar everyone here is with that. So essentially what this means is that we present an animal with stimulus. So we call this CS, so a conditioned stimulus, and we simultaneously present a US. And what the object of this type of training is to get the animal to learn that the CS predicts the US. So for fear conditioning, we use for odorant fear conditioning, we use an odor as the CS and a shock as the US. So those two things co-terminate. Um, and with the purpose of this is just to get the animal to learn that once you smell this odor, a shock is going to occur. And then the readout of this is percent freezing. So when a mouse is in a box and you shock it a bunch of times, it no longer moves. It doesn't run. It doesn't scream. It just sits there for some amount of time. So our readout, or our conditioned response, is that the animal freezes. So in this data from a previous study that was published from our lab, you can see that for the CS plus, so the plus identifies that it was paired with the US. You can see that there's a much higher freezing percentage than with unpaired odors. So these are just other odors that we presented, or versus just presenting the animal with clean air. And we see that difference stands alone, even if it's just the shock by itself, or even if it's just the odor by itself. And these are images that you'll see pretty commonly in my talks, where this is the dorsal olfactory bulb. And the explanation of what this picture is we look at populations of cells. So in this particular case, we're looking at, I believe, periglomerular cell populations in the dorsal olfactory bulb. And we mark these cells with a calcium indicator called GCAM6S, it's GCAM6F for fast, which allows us to monitor, so we can monitor the animal's response to the odor that's being presented before and after the fear conditioning, after they go through fear conditioning in a box. So in this case, the CS plus was methylvalorate. So before fear conditioning, you can see there's some activation here of the odorant, and it's specific to what odorant is being presented. So I know this is methylvalorate because there's a characteristic stripe whenever methylvalorate is presented. And then you can see the same thing on the right here after, but you can see the activation of this glom in particular is much higher. So we see an increase in activity after fear conditioning. So we have, once again, just to reiterate, we have our olfactory sensory neurons that are binding our odorant molecules. We have our PG cells that are affecting our incoming signal at the level of the first synapse from the OSNs to the mitral tufted cells. Then we also have our short stature axon cells that are dopaminergic and GABAergic. They're very complicated. We don't quite know what to make of them because they're both excitatory and inhibitory at the same time. But we're getting more answers about them as we move along. And we have the output mitral tufted cells, which makes up the olfactory nerve that then goes back to the rest of the brain to tell it about what's going on with the nose. So like I just showed you, we have odor cue fear conditioning invoking local plasticity in the olfactory bulb, that increase in activity that I talked about. And that's been published in both the olfactory sensory neurons, so the first, the axons of the first neurons to, touch an odor molecule, and also in the PG cells, which are those GABAergic interneurons. So my project primarily is concerned with um, determining what kind of information the olfactory bulb gets. So how does it know about something like a tail shock, which is somatosensory and not olfactory? Um, and that shocks actually evokes like an inhalation. So which is, if you think about it, can cause just a strong peripheral input that can cause activation. And it also could evoke some type of a neuromodulator release. Um, and so just leading to that. So as evidence of that central input coming down into the olfactory bulb. So one of my specific aims for one of my projects is to actually characterize this tail shock evoked activity in 4 different cell types in the early olfactory bulb circuit.
A quick question, Keith.
Yeah, go ahead.
In the chat, some Kashish asked, how strong or painful is the shock if it causes a freeze response? So
So typically the shock is about 50 volts when they're in the box. So it's different depending on how you deliver the shock. So there are 2 ways that we can deliver it, either on through a shock floor where they're being shocked basically through their feet, or through a tail shocker where the shocker is actually affixed to their tail and they're shocked. So in the case of that first study, the mice were fear conditioned in a box. So they're basically standing on a metal grate, and the shock is delivered across that metal grate at a specific time. And it's shocking at around 50 volts. And I know that sounds really high, but it's not. It's meant to be aversive and not like damaging in any way. So they're scared, but they're not in pain. They're not, there's no lasting pain that occurs.
so it's more like startling than like long lasting pain, right?
Yeah, yeah, it's more startling. So
Okay,
it's
thanks.
kind of it actually feels like if you've ever had somebody like play that trick on you with like the pack of gum where you pull it out and it shocks you. That's what it feels like, where it's like you kind of feel that little shock on your finger, but it's not anything that's too serious. But it's memorable because it has to be strong enough to be memorable, but it's not something that's going to cause lasting irritation or anything.
All right, thanks.
Okay. So my first bit of data that I want to show you guys is from the paraglomerular cells. So here again, we have that classic methylvalerate stripe. So this is just presenting the odor by itself. And then if we also shock the animal by alone, then we also have a different activation pattern that occurs, telling us that these two types of information are being processed somewhat separately. Then if we look at the traces, which are this is just a running record of the entire trial. So we have respiration here, and this is the fluorescence record. So once the odor came on, you can see, sniff, 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 sniff. So each peak correlates to an inhalation. And then you can see the same thing once the tail shock happens. So the mouse is startled, and we see this, the small first peak, and then this really, really large peak that you're seeing in the picture above. Um, so this is not just unique to the PG cells. This is also found in those short axon cells, those dopaminergic and GABAergic cells. And you can see the shock response looks a little bit different in those guys as well, but the methylvalerate stripe looks pretty much the same. And when you look at the traces, it also has the very similar characteristics. And if we look at the mitral tufted cells, which are the primary output of the olfactory bulb, we see another, the methylvalerate stripe and the shock response. And what you're looking at, it seems like a much more diffuse signal than any of the other cell types, because the mitral tufted cells are very deep based on the angle that we're looking at. So we're looking from the top down. So we're looking at the dorsal olfactory bulb, and the mitral cell layer is, as you saw, a couple cell layers deep. So the signal kind of scatters a little bit the deeper you go. Um, so this is just some of the evidence that I've gathered showing that there is some convergence of somatosensory information as well as olfactory information happening really, really early in the olfactory system. And it's not unique to shocks either. So this is an example of a mouse that got an odor, once again, a methylvalerate, followed by a tail shock this time. So this isn't the foot shock in the, this is with the electrodes affixed to the tail, and you can see that clear shock response. And then if we do a thermal stimulation, say on a hind paw, then we see a similar response in the olfactory bulb that seems a response similar to that of the shock. And if we present something like CO2, which causes kind of a burning sensation in the nose, we also see these really interesting signals that occur. So this kind of leads to a second project that I'm working on that's kind of new, where we're looking at how much information the olfactory bulb receives, right? So we have the shock from the tail, that's one area of the somatosensory system that's getting a signal all the way to the olfactory bulb when it happens. Then we have the thermal stimulation, which is taking advantage of a different processing center, somatosensory processing area, and we see a similar response. And then if we present CO2, which is a trigeminal irritant, then we also see that response. So it's really interesting to see that the olfactory bulb not only processes olfactory things, but it also is affected by external factor, like many external factors that have to do with somatosensation or trigeminal stimulation. And that's all I have. So with that, I would just so that's the project I'm currently working on. And now just a little bit about me, just in case you guys have questions about my background. I um I did my undergrad in down in New Orleans at Southern University at New Orleans. And while I was there, I did research on environmental microbiology, where we looked at the effect of the BP oil spill. Um, looked at the effect of the BP oil spill on some of the shrimp species in the Gulf of Mexico. Then following that, I did research looking into the auditory system of frogs. So we did extracellular recording in the inferior colliculus of some frog species, and just to basically get tuning curves, which are how loud does a sound have to be at what frequency for an animal to hear it, which hadn't been done for the particular species. It's called Ophagopamilio, or the poison dart frog. And in addition to that, that summer we also worked on another project that's kind of similar to what I'm doing now, where we looked at peripheral changes in the auditory system. So changes uh, in the sorry about that. So changes in the inferior colliculus as a result of stimulating the thalamus, which is just one level above the inferior colliculus in the auditory system. So just kind of showing once again that there can be meaningful changes happening very early in our sensory systems. Then I've also done work on the visual system. So that study was more neuroanatomical, where we did a lot of sectioning and taking pictures of retinas and measuring density of rods and cones and focal distances by looking at the lenses, the distance between the lens and the retina. And that was also in frogs. So we did kind of a comparative model between nocturnal and diurnal frogs, just to see like what makes an eye sensitive, and how these animal species evolved, how their eyes evolved to diurnal versus a nocturnal environment. Um, and finally, I did work with humans, where we studied the expected value of control theory, which essentially is an equation that predicts whether or not you'll make a decision based on reward and cognitive cost. So if something is way too cognitively costly and the reward isn't worth it anymore, then you'll give up, right? You'll hit a break point, is what they call it. Um, so I've done those are the research projects that I've done before I came to Rutgers. So I think that's all I have right now. So if you guys have any questions or anything, I'm here to answer them.
In the chat, I think Ayan and Luna were talking about how this research like relates to PTSD and how patients get habituated to stimuli that trigger them. I think, and then Luna asked if there was like CBT.
So the first, so yeah, so this is very closely related to PTSD because in patients who have post-traumatic stress disorder, we definitely see sensitization of sensory sensitization. So they get it's called hypervigilance, where they have where their senses are very heightened, and whenever they experience a similar stimulus, so just like we had with our, when they experience a CS, then they're instantly transported back to that aversive event, to that traumatic event. So hopefully by understanding how this circuit works and how information, and looking at how we could manipulate this circuit, this early circuit of convergence of information about shock and odor, to help us out, it would be yeah, it would be good for that area of research. But I'm still working. And what was the second question? Sorry.
The second question was just about CBT, but I think you answered that.
Okay.
Um, okay.
Um, could you possibly go into like your could you go into what kind of led you to go down the path of creating like the mind network, and what kind of experiences led you to that, and how you think people would benefit from it, since we're collaborating on a program, right? I think it'd be cool if maybe some of the members got like had an idea of what they're getting into.
Yeah, sure. The Mindneck. So I went to a really small university at Southern University at New Orleans, is a satellite campus of Southern University. So um, mentoring kind of influenced my early career very much. So I started out as like a work study with one professor, and that kind of snowballed into my first research project. And when I came to Rutgers, I noted like you guys are a much bigger school, right? So it's a lot harder for you to have meaningful relationships with the professors you come across. So it kind of got me to thinking that, even though, it's not possible to have everything all in the same like academic space. So like the reason I was so close with this, with my first advisor, was because I had taken classes with him, and he knew me from around, and then we got to talking about research, and he told me about programs that were around. So because I benefited so much from that relationship, I felt that it was something that we could bring to Rutgers, but it would have to be separate from like classes and things like that. Um, and so that's kind of what it was. That's why it was created in the first place, is just to give students that, the support that they need throughout their careers. So, understanding that, small things like I need to get into research earlier rather than later, asking questions about classes that you're taking. So the relationship can look, however you need it to for you to succeed. And we also make sure that we don't have, so a lot of other mentoring programs typically are for, students who only want to go to graduate school, or students who only want to kind of go into, the business side of things and industry. But our goal is just to give you whatever support you need. So if you need a mentor to tell you about, how to get an industry position, we'll have that. If, depending on how many other people are interested in industry, we can have speakers that we bring in speakers based on, what you guys need. If you will take votes periodically on, what kind of speakers everybody wants to see, and we'll bring in people based on, what we see. In addition to that, also, like, so that's kind of the purpose of the mentoring part. But then another experience I think helped me a lot as an undergrad was whenever somebody brought in a speaker, because the school was so small, I got a chance to actually sit down with some of these great minds and just have, fun conversations. And it helped me get past that barrier of like they know so much more than me, and instead and help me to get more comfortable with like addressing per faculty, outright, and just like, tell me about, what kind of research are you doing, and not being afraid to ask those important questions about whatever, research that they're doing, or how they got to where they are, because if that's somewhere that I want to be, any information that they can offer me is valuable. So I felt that's also another part of this that could be really beneficial, is just being around a group of people who all study similar things, um, and you can ask any questions you need to, and you also have that support because they've probably been, in a situation similar to what you're going through at some point in their career, and can kind of help you through that, even if they're not your direct mentor, right? Just having that network atmosphere and having coming together as a group can have a good, I think, a good effect on your experience as a student.
Yeah, I've actually I was at one Mind Network talk. So
Mhm.
I actually was with Dr. McGann was like across from me, and it was like a very like intense cerebral discussion about like the auditory system, and I didn't understand a single word, but I knew
Mhm.
I liked what I was hearing. Um but I think my other question is, in your opinion, apart from kind of surrounding yourself with mentors, so you can gain more perspective and make it seem like a more approachable and achievable goal to be where they are.
Mhm.
Um, since it's encouraged for people to get involved early in research, what kinds of things would you recommend doing, like in terms of I guess catching up to speed with terminology, especially in academia, because it's often so dense and so hard to navigate, especially with neuroscience. uh, so yeah, would like journals be the best way to do, like reading through journals, or would like looking up YouTube? Like, what was your approach, like in kind of better understanding terminology, at least early on?
So for me, getting acclimated to the terminology in the field was, so because of those early experiences, and because I broke the ice with so many professors and got comfortable talking to them, I was definitely privy to many conversations that were way over my head. And what that did for me is it caused me to go out and, yeah, Google search things. I know this professor's name, and professors love talking about their experiences, right? So one of the things I was never afraid to do is also ask as many questions as I could, because they love talking about their research. And if they see that you're like, an empty vessel that they can pour their knowledge into, then they'll be very glad to, break it down to the most elementary level, just so you understand what's going on. And through a lot of those experiences, talking to a lot of faculty members, talking to graduate students, and just forcing myself to be a part of those uncomfortable conversations, until they started making, I was able to learn the terminology kind of passively, and then, trying to be as active as possible in the conversation, even when I only understand small parts of it. I think also was helpful. So like you were saying, like, Dr. McGann was probably talking on the highest level that he could think on, which is way above even where I'm at right now, right? So that can be scary as an undergrad. So, but you knew he was talking about the auditory system, right? You knew, I'm sure you recognized a few of the terms that he used, if not all of them. So just take none of them. You didn't get.
I got some of them, but not all of them.
Yeah. So I mean, it's that's fine. But yeah. So asking a lot of questions, and just, it's uncomfortable at first. It's very uncomfortable because you feel like, I feel like I felt so dumb at first. I was just like, I should know stuff like this. I'm an undergrad, like I'm taking classes and stuff, like what am I doing with my life? But through a lot of that, trial and error, and pushing myself past that, well, I'm an and forgiving myself by saying, yes, I'm an undergrad, I don't know some stuff and I want to know stuff. So just asking the most, the most fundamental questions will eventually get you to understanding how these things work. And what else it'll do is it'll prepare you for graduate school, because all we do is critical thinking. So, asking all those questions and stuff at the time was just a part of me, but over time I saw that my ability to think about their projects and how they progress that project, and progressing the ideas that they bring up in those conversations also became a part of it. Because naturally, once you get more comfortable in these spaces and talking, then you'll be more comfortable sharing your opinion. And even if it's an opinion that's that's far-fetched, they'll explain it in a very eloquent way how wrong you are, right? And then that'll also be a way for you to learn some new things as well. So yeah, so that's what I would say as like how to get up to speed, is just asking questions, talking to people. Of course, reading, I would never tell you not to read. Reading is always great. But a lot of times, if you don't understand the terminology, reading can be frustrating. If if you kind of dive into the deep end of a publication and they're just full on terminology, then it'll be like I can't even follow like what they're doing, and it's just frustrating, and you'll put the paper down and like it won't be that helpful. But after you have a few of those conversations, and even talking to some of your professors that teach some of your classes, right? Looking at what research they do, and just like, hey, can you tell me more about, you were talking about the auditory system, but I know you study the olfactory system. So, how are these two systems similar? How are they different? And then he might get into terminology, and if he gets too far above, just say, hey, wait, come back down here. I'm down here right now. So yeah, I think that's a good strategy, and that's kind of what I did. Is just ask questions and things like that. So
Yeah, that's good advice. Especially the part with I think that's also really beneficial because I know I had a friend who I took a seminar with, like a neuroscience seminar with, and he would go to the Library of Science and Medicine in at Bush, and he just binge papers. I'm like, first of all, how are you doing that? And second of all, like, yeah, like I didn't find it productive to just brute force, right? So I think it's
mhm,
actually really a really good thing that not only does this benefit you now, but it benefits you in this, I guess in like a more abstract sense, you're learning, but you're also learning how to learn, right? Like
exactly.
especially in the graduate setting, which is I
Mhm,
think super super important.
right.
Um, does anyone have any questions? Because I want to ask another one, but I don't want to like take up the floor. So
I'll ask a question. How do you get more involved in the my network? I'm not really familiar with it for myself, so I wasn't sure like how we can get more involved.
So I think I sent you guys the link. So so you can send us an email to to our network email address, and and just kind of, if you have any questions about anything, you can send them there. Give me a second. Let me see if I can find the email address. But that's one way that you can kind of get in touch with us. And then if you want like a mentor, then we'll send you a Google form to fill out that'll tell you more about, that'll kind of ask you about your background and help us kind of pair you with a proper um, mentor.
I think you can also, if you take a look at, if you want to sign up, you can, like, our sign up form will probably lead you back to the same, because I think it's all in the same kind of network, because we're utilizing the network anyway, so it all goes to the same place, right?
Mm.
Um, but yeah, just wanted to
Yeah.
Yeah,
mention.
So I just posted the email address also in the chat for you guys.
Anyone else
yeah,
have a
we have
question?
the link on our Discord server and our social media as well.
Yeah, and also if you have, yeah, so that's where you can send any questions you have about joining. But um we're very open to any like students in need. So so it's not going to be anything like a very serious time constraint. We usually have like 2 events a semester that involve the talk. So talk about science, tell us about your experience, as a person in science. then we break out into smaller sessions so that you can have that FaceTime with the speaker and also FaceTime with some of the other mentors. And if you have a really, really great conversation with somebody at one of those events and you really want them to be your mentor and they're available, then you can even send something to that email address and tell us like, hey, I met such and such at your last event and I would like to be a part of this. And then we'll, send you the same form and we'll notify them that, they have there's somebody that wants to be mentored by them and we can go from there and it'll kind of expedite the process a little bit of sorting and matching people. Yeah, so
Okay, I'll ask my question.
mhm.
So, uh, is I know the Mind Network it stands for um I might not
So
know, but I know it has neuroscience in it. I think it's minority. Is it minorities and neuroscience? Is mentoring and neuroscience? Can you refresh me?
so we actually kind of 86. So we recently expanded to other areas of neuroscience. So we're just going to be the Mind Network. Instead, we don't. It no longer stands for anything.
Oh, cool. Okay, so
Yeah.
I don't
So
know.
because it was a little bit restrictive because it was specifically talking about people who study neuroscience, and now we're more concerned with anybody who studies the mind, whether that's clinical psychology, social psychology, Rux, behavioral, anybody who likes studying the mind in some capacity, whether abstract or physical.
Yeah, that was pretty much my question. Like, are you still mostly focusing on neuroscience or other fields? That's great.
Yeah.
And my last question. I'm sorry, I'm like barraging you here.
All right, go ahead.
My last question is, how are the Mind events working virtually, and how are like mentoring meetings working virtually,
Uh,
if they
so
are at all?
so the mentor meetings are usually not they aren't that affected by the virtual environment because so we asked that our mentor meet like well before corona we asked that they meet you in person and you guys go out to lunch or something just to kind of help that relationship along so they're like a person and not just the email address or phone number or something. So using Zoom and stuff like that isn't that different for the mentor meetings themselves. I can't speak specifically for every mentor mentee pairing as far as difficulty, but I haven't heard any complaints or anything yet about being able to meet with mentees over Zoom or anything like that. And as far as our meetings are concerned, so we haven't actually had our first virtual meeting. This will be our first semester, like with a virtual meeting, because I was busy with like my qualifying examinations last fall, and everything was happening weird over the summer and stuff, as you all know. So we're looking to have our first virtual event this spring, sometime in March or April. Of course, I'll send you guys information if you guys want to come to that. And so we don't know yet. We're working on getting. We're trying not to use just the Zoom breakout rooms and actually find um, things like this program called Spatial Chat, where for the breakout session afterward, which will be an opportunity for you to kind of move your icon around the virtual room and talk to people because we don't want to lose that. That's a very important part of our events, the kind of in-person mentoring that occurs at the event itself. That's like independent of whether you're paired with somebody or not. It's just like you can. That's like the strength of the network itself. So we're trying to find a way to bring that to the virtual space, but we don't have an answer quite yet, but we will hopefully soon.
All right, great. Thank you so much.
Mhm.
I'll ask again. Does anyone have any questions?
Boring logistics question. Which Discord channel has the
You broke up, and I didn't hear anything you said. I think I heard Discord and mine. That's it. Oh, are you typing it? You're okay.
Is it which Discord channel has it? Okay.
Oh, okay.
It, it
Okay.
might
So
either be in general or broadcast, right?
it's in broad. Yeah, the information for this meeting is in broadcast. There was a brief glitch that Luna pointed out to me actually. That the broadcast channel wasn't viewable to the general members for like a hot minute, but I think that's fixed. I think we're good now. So some more of the information is there. Um, if you want to sign up, I have the link ready. That's not the link. Hold on, give me a second. Here it is. I have the link to the form ready. Here it is in the Zoom, and I will also be putting it in the general chat for anyone to access if they want. And yeah, if you have any questions, Keith, would you be okay with providing like your email or something if anyone wants to reach out to you, or
Oh yeah,
would there
sure.
be a better way to reach out to you for that?
Yeah, my email is fine. I check it pretty regularly.
All right, cool. Um, yeah, if anyone has any questions, you can reach out to us, or more in-depth questions about the Mind Network, you can reach out to Keith. Um, if there are no more questions, I'd like to thank Keith for coming. This is super informative, and um, we really appreciate you letting us use this really amazing resource to teach our community. It's really great. You know, it's a really good resource for us. So thank you everyone for coming. We really appreciate you taking your time on a Wednesday night to be here. Um, and yeah, see you next week for our next meeting. Bye, guys.
Bye, y'all. Thank y'all for having me.
Bye.
Thank you.